a renowned speaker for our webinar. As everyone knows, genetics is one of the high priority areas in autism research, and today's presenter is Dr. Matt Fletcher from Autism Speaks. Dr. Fletcher is the Vice President for Genomic Discovery for Autism Speaks, and is going to be speaking to us about the use of genetics to help improve autism research and care. So I'll turn things over to Dr. Fletcher.
Great. Thanks, Dan. Thanks, Audrey. Really appreciate the opportunity to come and present the work that we've been doing in this area at Autism Speaks, and to just sort of give a high-level view of sort of why we've invested in this area and what we hope to see come out of it in the end to really advance care for individuals with autism. So the main focus, or at least what I build a lot of this talk around, is our missing project. And what missing is, I'll just wait, let me make sure. It looks like my screen, can you, I'm sorry, it looks like the sharing isn't working right. Audrey, can you see did it look like my slide moved ahead?
No, it did not. It did not advance.
Okay. Yeah, it says the screen sharing is paused, so let me see if I can figure this out real quick. Okay.
Okay, now you're, yep, there you go.
Okay, sorry about that. Technical difficulty. Okay, so anyway, so start out, sort of the talk is built around the work that we've been doing in this area, primarily with our missing project. And what missing is, at a very high level, is it's this effort that we've undertaken in collaboration with Sick Kids Hospital in Toronto and Google, now Verily Life Sciences, to sequence 10,000 whole genomes from families with individuals with autism, and to make that data as broadly available as possible through cloud computing resources. And that really to fully embrace the concept of open science, to make the best use of this data, recognizing that the complexity of what autism is at the genetic, at the biological, at the medical level, really to be able to fully understand that, to make the best use of this data to help crack that code, it really is going to require as many different people from as diverse background as possible to really be able to engage in this data. And that's what we've tried to establish through Missing. One of the advantages of having something like cloud computing is the fact that we can take these very large data files and make them immediately available to folks anywhere in the world. And then to try to build tools around that to allow anyone, regardless of their expertise, to be able to engage, search, utilize that data to further their own research and understanding. As far as where Missing stands right now, where this overall effort is, and what Missing actually is in real-world terms, at this point, we are sharing through the Google Cloud Platform 5,204 genomes. These are what investigators who have applied for and received access to the missing data currently are working with. And this really is the largest publicly available data set of whole genomes anywhere that researchers can make use of. We have another over 2,000 whole genomes that actually we've completed sequencing on, and we're getting ready to release. Part of this is that for a subset of these, we're actually going through the process of making sure that, in fact, all of our informed consents are in line and that they're fully approved from an ethics legal basis before they get shared out to the broader research community. But we anticipate another 2,000 or so coming out within the next month or so for investigators to work on. And as far as that research community that we've engaged, we have now actually over 100 different researchers who have applied for and are utilizing the missing data now. These researchers are in, at this point, 7 different countries and represent roughly 40 different institutions. So it really is a worldwide community that we've been able to who've been able to tap into this data so far. Built around this data are some new tools that we've actually put in place to make it easier to work with the data. One of them is represented by some of the screen grabs at the lower side of this slide. And this is just a web-based portal that we put in place. So anyone who has access to the data, if they don't have, really, a great deal of expertise in bioinformatics and be able to build their own algorithms, be able to dig into the data, well, you don't need that, because through the portal you can execute very basic searches to be able to find information that you might be looking for. So if you're interested to know whether or not within this database there exists any variants in your favorite gene, you can search by gene. You can filter based on whether or not those variants are predicted to be deleterious, whether it's a nonsense versus a missense mutation. You could search by individual sample if you're looking for a particular sample that exists within the database. You can search by genomic region. You can take all this, go all the way down to the individual medical data. So that way, again, if you're interested in variants in the gene SHANK3, you can see what's there and see what actually characterizes that mutation from a medical perspective, at least within this data set, through just simply by browsing through this web-based portal. And again, the data is being used. Even though the majority of this data has only been available to researchers for about a year, there have already been 11 publications that have come out utilizing the missing data set. So we are seeing, in fact, that the data is being used, and it is driving new knowledge in this space. And in fact, we're very excited about the most recent of these publications, which I'm going to come back to, which was a publication in Nature Neuroscience about a month or so ago that really did the first in-depth analysis of those first 5,000 genomes. As far as where we're going next with this missing data set, a couple of different activities that we have going on. One of them is around trying to increase the utility of this data set to an even broader audience than we already have done. And this is through a collaboration with the Global Alliance for Genomics and Health, particularly Mark at DNA Stack and Serena at Elixir. And what we're working with them is to make use of what's called the Deepen program that the Global Alliance for Genomics and Health has set up. And what this does is essentially it provides a catalog of the variants identified in different data sets around the world, and again puts that in a very easily searchable database. So right now within the missing database, we have certain limitations on both how the data can be accessed and how it can be utilized. That goes all the way back to the original informed consent that governed this data from the participants. And that includes the fact that because the missing data certainly could be, even though we've de-identified it, there is the information available that if someone was so motivated, they could re-identify individuals. And because it does include it does include personal data, personal medical data, there are certain safeguards as far as the process people have to go through to access the data. What the Deepen program does is it really strips away a lot of that potentially identifiable data. You take away the phenotypic data, the medical data, and you provide just a list of the total variants that are found in the data set as a whole and the percentages. So it's no longer individual genomes, but it's a collapsed data set. And the thing is even with stripping away that data, it still is incredibly useful for the community at large, especially for those who aren't even asking autism-directed questions, just to be able to use this as a reference set, to know whether a variant they're finding in their own data has ever been found in any other large genomic study. And so this platform allows us to push that data out into that broader audience and to make it immensely shareable and to allow those other genomic researchers to use this as really a reference data set. And so we're working through that process. Additionally, one of the other initiatives that we have going on is a collaboration with a company here in Boston called Genospace to build out what we're calling a community portal. And I'll come back to some of the principles behind this later in the talk, but essentially, one of the promises that we made when we first launched Missing was to the participants themselves. And again, I think as an organization, that sole purpose really is to represent and to benefit families. We felt it was very important that if we believed that this genomic data had value, and obviously we do by investing in this scope of study, that those families should be able to benefit from this data, not sometime in the indefinite future, but now. And so what we're doing through the community portal is building an avenue for those families to be able to actually retrieve their own data, give them the tools to at least gain some understanding of what was found within that data, and then to be able to take that information in a format to be able to share that with their physician and to engage in a discussion about what that could mean for their own care and their own path, their own travel, their own journey with autism and its diagnosis within their family. Additionally, one of the other tools that this sets up, this portal is meant to set up, is to allow families to connect and to converse with each other, particularly around their genetic diagnosis. And again, this is a theme I'm going to come back to multiple times within this talk, but the idea that what we're finding again and again around these genetic subtypes of autism is that there's value in this for the families in the type of information that they can gain from it and the type of conversations they can have. That the kind of questions they have around from a newly diagnosed family about what's in their future can be, there's likely to be much more value in the answers they get from the community if they know that they are within the same genetic subtype. And so the portal is meant to be set up to allow those connections to happen and those conversations to happen. And again, I want to come back to this in a bit and in a sense harp on that a bit more later on. So now sort of zooming back out again, as far as why go down this path? Why make an investment like this? Certainly, there are so many unanswered questions in autism. There's so many places that an organization like us, like Autism Speaks, could go that we could make this investment. And certainly, one of the things that we thought about before launching this program is really where is the greatest opportunity for us to have the largest impact? Where is our investment most likely to return something meaningful in the near future? And I think, again, one of the things that keeps coming back again and again is the role of genetics in autism and the kind of information that we can gain from that. That, what we really understand is that there is a substantial genetic role here, and that by making investments, by generating a data set that does not exist up to this point and making it broadly available, this provides us one of the best opportunities to really speed those new discoveries, to accelerate our understanding in this space, and to really drive autism care and diagnosis in a better direction. And so that was sort of some of the rationale behind it. In fact, this has been underscored again and again by study after study as we continue to understand autism from the perspective of genetics. There was a couple of papers that came out at the end of 2015 that just underscored this point, that essentially what they said is that based on our current understanding of genetics role in autism, that, and then again, just based on our current understanding, that you could ascribe 40 to 50% of autism cases to rare, highly penetrant mutations. And so there is an ability to be able to gain a greater understanding, greater description of a significant portion of autism with more data in this particular area. And of course, the other decision that was made at the outset was the idea to pursue whole genome sequencing. Because certainly one of the, I think right now, you talk about in the diagnostic lab, the sort of standard is to do a microarray evaluation as far as the first level of genetic diagnosis. In the more research area, most of the work right now is done in just sequencing the exomes, the part of the genome that contains the genes, the part of the genome that transcribes RNA to protein. But that's such a small amount of our genetic makeup that, in fact, in sequencing the exome, we're only looking at roughly 1 to 2% of the DNA that makes up who we are, that controls and regulates the cell's gene expression, and that in doing so, we are likely missing a significant portion of the biological controls that relate to the causes of autism. So if we're going to build a database that we hope to be useful not just now but well into the future, why not go ahead and invest in getting the other 98% of our genetic code here? So why not do the whole genome, especially now that we've got this nice intersection of capabilities, technological capabilities, and cost. It does not cost significantly more to do whole genome than it does whole exome. So let's capture that. And even if we don't fully understand how to interpret the non-coding portions of the genomes, the part of the genome that doesn't contain the genes, well, the only way to really be able to build those tools to gain that understanding is to have the data in the first place. So just in that sense, it's a bit of a chicken and an egg thing. That certainly some would argue why do the other 98% if we can't interpret it? Well, we're going to continue to have trouble interpreting it until we have the data to begin to build the tools to build that understanding. So with the missing data set, hopefully we're providing that raw material that will drive the innovation, that will drive our understanding. And in fact, you're already seeing reports come out where people have taken some tentative steps in this direction. This is a paper that came out again early last year, out of Evan Eichler's lab, work done by Tashelle Turner, where they began to look at these non-coding portions of the genome. And in fact, sure enough, they find variants, find changes in the DNA in these non-coding portions of the genome that they believe contribute to an individual's risk of developing autism. And so, in fact, already we're finding clues to the biology of autism by looking in this other 98%. So again, with the data out there, we expect more of this kind of work done and a greater appreciation of, in fact, that this part of the genome that has been called junk DNA actually isn't such and will play an important role in that understanding of autism. So what is it that we hoped to get out of this? What were the discoveries? What was the movement in autism treatment that we hope to see as a result of producing this kind of data set? And so now I return back to that Nature Neuroscience paper I mentioned before, and it exemplifies, I think, sort of in some ways what you might say the low-hanging fruit of an investment like this is, and that is just a greater expansion of those genes that we could draw a direct line between their function and a diagnosis of autism. And in fact, that's what we're seeing. That what you're seeing on the right-hand side of this particular slide is a table from that Nature Neuroscience paper that lists a number of genes in which we were able to find within the missing data set variants that had a strong association with autism. And in fact, out of the data set, we were able to identify 18 genes that had previously not been associated with autism, or in other words, that this paper was associating with autism for the very first time. So with more data, we are in fact gaining new understandings. We are finding new genetic subtypes of autism, and we continue to expect to find more of these genes with more data. So that, of course, is useful, and that is a first level of what we want to see. Additionally, what you're also seeing again and again out of these kind of genetic studies is that, in fact, these genes often relate to each other. They're often found in the same molecular functional pathways. They often carry out the same type of function within the cell. In this particular case, again, this is on the left-hand side of the slide is a figure from the Nature Neuroscience paper where taking the genes that the 60-odd genes that we identified within the missing data set and doing a pathway analysis, you could find 3 primary functional domains in which these genes operate. Not surprisingly, one of these clusters is around neuronal and synaptic function. Another one is around RNA processing. And then finally, transcriptional regulation, how genes are turned off and on, when they're turned off and on. Again, not terribly surprising in a developmental disorder that genes that regulate when other genes come on in development would be perturbed in something like autism. But, and so this does, again, give us a better understanding of the underlying biology of autism. It also helps set up the idea that, in fact, you are seeing distinct functional, biologically distinct types of autism, at least in their underlying causes. But then also, whenever we want to take a step back and think about the development of new therapeutics in the autism space, potentially this is also going to be very useful. That, again, the idea that even though we're finding hundreds of genes, potentially, if you look at this from the pathway level, you could think about now within this that certain genes all function in the same area, so autism that arises from changes in that pathway could all be amenable to the same kind of therapeutic intervention. And these genes themselves suggest what those therapeutic interventions could be. A number of these genes already are found within pathways or make proteins that we know can be targeted with small molecules, with chemicals, with drugs, and therefore are set up as good potential drug targets. And there's even a subset of these already are known to be targeted by small molecules. And for some of these, in fact, there are already animal models that exist, so we could even think about testing whether or not any of these molecules that interact with genes that we know are involved in the biology of autism, whether or not that targeting these genes are going to have a meaningful impact on the outward manifestation of these genetic changes and on autism itself. So it does begin to think about what that next step looks like, and really this information helps power that step. Additionally, one of the and again, along the lines of thinking about what that next generation of interventions might look like, another interesting observation that's made in this data set, and has been made before as well, is that in sequencing families, which we've done in this, most of the samples we've sequenced comes from trios, a mother, a dad, and an affected child. You get a situation in which a change, a mutation, was present in both the child, but also in an unaffected parent, and a parent that doesn't have a diagnosis of autism. And what this sets up is the idea that potentially what that parent has is a compensatory mutation, that they have an additional genetic change that has resulted in them not manifesting an autism diagnosis. And if we can understand that compensatory mutation, it sets up a way of potentially developing new therapeutics. And to give you a better idea of what I mean in that, there's a nice example that comes from the work in HIV/AIDS. That really one of the key findings that helped power development of new therapeutics, specifically the drug Maraviroc, was the fact that there was a population of high-risk males who are continually exposed to the HIV virus who were not getting infected, who weren't showing infection with the HIV virus. And what they were able to find is, in fact, they carried a mutation in the gene CCR5 that prevented that infection from happening. It changed one of the key receptors on immune cells that HIV used to infect cells, and that prevented the infection in these individuals. So by producing then a drug that replicated that mutation, you had an effective therapeutic. So in the same sense, if we could find what change existed in those parents that potentially compensated for a mutation that otherwise would have resulted in autism, that in itself could give us important clues on what a next generation therapeutic could look like. Finally, one of the other things that this data does, I think, is provide a bit more clarity even when it comes to diagnostics. And this goes to both to the clinician and to the families themselves. I mean, on sort of the clinician scientific side, having this level, this granularity of data is incredibly useful in understanding what that genetic diagnosis actually is and what it could mean in thinking about the downstream consequences and trying to utilize this information in thinking about care. So here's one example of an individual who had been genetically diagnosed by microarray. And what you can see is, by that red line on the bottom, is what the deletion was that was predicted to exist with this individual based on the microarray. And again, with the microarray, you're only testing limited spots along the chromosome. So it did say there's a deletion here, but it gave this huge range on the X chromosome where it said it was deleted. Now with the genomic sequence, you can see that in fact that actual deletion was less than half the actual size. And so whenever you want to try to understand from a genetic counseling standpoint, or even from trying to translate this finding into an understanding of medical risk, this has a huge impact, right? I mean, you're talking about the difference in hundreds of genes either being present or absent. So having this kind of clarity is incredibly useful. And again, from the patient side of things, one of the things we hear again and again is, even if there isn't a direct step to be taken in the medical management of autism based on a genetic result, just having that clear genetic result is useful, it's empowering, it's meaningful to families in many cases, because it does provide some kind of answer, it provides them with some kind of understanding about what's happening within their family, within their child, within them themselves, and thinking about what that looks like and what that means to them. But so all these things are sort of real, tangible, and certainly part of why we want to make an investment like this in missing. But sort of bigger picture, what we want missing to be able to power, or to be at least be part of, to help make possible, is the movement away from what really is an umbrella diagnostic term of autism spectrum disorder to something much more specific and much more meaningful. The idea that really within that diagnostic term, you include such variability in outcome, in medical needs, in development, in potential therapies and treatments, that it's not particularly useful. It's not particular to the researcher trying to design studies. It's not particularly useful to the physician trying to decide on the best clinical care. It's not useful to families to help inform them on what's in front of them. And if we can begin to better segment, to better understand the subtypes of autism, that could potentially be one of the more meaningful impacts in actually advancing autism care forward. Just to underscore this idea of autism being, an autism diagnosis not being the same from person to person, often it's not even the same within families. This is a figure from actually the very first paper to come out of missing, where they are looking at some of the families where we had multiply affected children. And in that case, in many of the families, the genetic subtype of autism was often different between the two children. So even within the same family, you weren't talking about the same type of autism. So now you want to expand that across the population. You think about just how different it is, in the idea that really each individual needs to be considered separately and not by this single term. There have been efforts to try to segment autism into different subtypes. And this is just an example of one a study done here in Boston by a group, I believe, at Harvard, where they've taken thousands of electronic medical records for individuals diagnosed with autism and did a principal components analysis and found 4 different groupings of individuals that they felt could distinguish, that you could distinguish these 4 different types of autism based on their combination of associated medical issues. And so you have one that's more defined by developmental issues, one that's more defined by seizure disorder, another one that's more defined by anxiety and behavioral issues. And once you're in, and these subtypes do provide useful information. If you were to assign an individual to one of these subtypes, it provides information on the likelihood of any of these comorbidities developing, some kind of timeline on when you're most likely to see these comorbidities begin to present themselves. And you can begin to think about a more forward-looking type of care for these individuals, because you can make some assessment about what they're most at risk for and what you should be focused on, instead of just this general idea of all of the potential comorbidities associated with autism. In fact, I think this kind of thinking, just to underscore why this thinking I think is so important, is just, again, another study from 2015 where they reported the lower life expectancy of individuals with autism. And to quote the conclusion from this, the premature mortality was markedly increased in ASD owing to a multitude of medical conditions, which again just underlies the fact that likely we're not providing the appropriate medical care for these individuals, and that in turn is resulting in a increased mortality. And even with the kind of classification we were talking about in the last slide, part of the issue of a classification based solely on phenotype is the person can only be assigned to that classification based after the phenotype has arisen. It can't be done ahead of time, and ideally you'd want to get in front of these issues to be able to begin to do preventative care before the first issue arose, to limit the impact it's going to have on that individual. And I think that again is where genetics comes into play. This is just an example of one of these genetic subtypes. This is CHD8, a gene that was first identified in 2014, where whenever you take out from the general autism population those individuals who have CHD8 mutations, you see that in fact these individuals, just in the picture you can see they have a very similar craniofacial structure. What's been found is they have a similar developmental path. They have similar, they generally across this particular genetic subtype have increased risk of epilepsy, schizophrenia. That you can say things about, you can provide an understanding of the medical issues and needs of this population with much greater specificity now that you have that information, the genetic cause, than you could before, whenever it was just autism spectrum disorder. And I think that's very useful. And then again, just to highlight again, the value this also provides the families is that with this identification of this gene, we've seen families form communities around it. This is an example of a Facebook group that is formed around CHD8, where these families can come together and talk about it, where again, now a family who has a recently diagnosed child with a CHD8 mutation can ask of a family that has a 13, 20, 35-year-old with that same mutation what their life experience is, and their answer back is going to be likely much more meaningful, much more informative than if they ask that same question of the general population. Again, this is just another example of a syndromic form of autism, in this case Duke15q, where after the identification of this particular genetic perturbation, that in fact there have been centers set up that specialize, that focus on the medical care and issues of individuals with this specific variant, have come to understand what specific needs are associated with this particular variant. And in fact, again, this is a population that they know has a high rate of seizures associated with it. So as a result, now this group of clinicians that have focused on it really thought about what the best standard of care is for people with a Duke15q CMV, recommend that upon that diagnosis of Duke15q, the individuals undergo a 24-hour EEG evaluation to understand that seizure risk, to be able to get in front of that. Again, knowing how damaging and the lifelong impacts of uncontrolled seizures can be in the individuals, to now have that information and to take that action, to do that evaluation right away, and ideally be able to intervene before that first seizure, very significant seizure ever happened. And just a final example here about thinking through potential opportunities based on this knowledge is a mutation that was identified in a number of families within our missing data set. This in a gene called period. Period is part of the circadian clock pathway. These are the genes that set our internal clock, when we're sleepy, when we feel like waking up naturally, when we're hungry. These are the daily rhythms that are set up within our body. They're controlled by the pattern of genes that cycle off and on in 4, 8, 12, 24-hour periods. And period is one of the genes that help regulate this. Of course, again, I'm sure as everyone on the phone knows, sleep disturbances are a significant issue with individuals with autism. And the relationship between sleep disturbances and behavior, especially the behaviors best associated with autism, are fairly tight. In fact, you could think even for yourself about the effect of sleep disturbances on you whenever you don't get enough sleep and what that means to your behavior and your attitude. And then you compound that with the other issues of autism. That in fact, it's been observed that whenever you can provide good care for sleep disturbances, that in fact you do generally see improvements in behaviors associated with autism. So now, what does it say if, in fact, for a subset of these individuals with autism, one of the genes driving their diagnosis is actually related to the regulation of sleep and sleep cycle? Could it be that for these individuals, the most impactful treatment could be on actually dealing with the sleep disturbances? And I think these are some of the things we'd love to be able to explore as we go forward, is whether or not a more directed type of intervention could be developed based on understanding what the most important variations, genetic variations, are within that individual's diagnosis. Let's see what time. Okay. And thinking about genetics and segregation of the autism population even extends to thinking about the development of those next generation therapeutics. Again, something that's been proposed as potentially beneficial for individuals with autism for a long time has been oxytocin, the so-called social hormone. And it has been tried in trial after trial after trial with highly variable results. Some folks report a very positive finding. Some studies show no real benefit at all. And I'd say that part of the issue has been, and has always been, and will continue to be, in drug trials involving individuals with autism, is the failure to properly assess and segment your trial population based on their genetic heterogeneity. That goes to both the underlying cause of their autism. Again, going back to that slide I showed around pathways, where in fact you may want a very different therapeutic for those individuals who have deficits in genes that control synaptic function versus those individuals who have deficits in genes that control RNA processing, but to the actual gene that your drug is targeting. In this case, there's a study out of a group in Japan that reported that, in fact, variants in the oxytocin receptor itself had a role in predicting outcome for the use of oxytocin. So potentially going forward, if we better identified the population most likely to benefit from this, you could actually be able to find that, in fact, oxytocin could be very useful for subpopulations. We could identify that subpopulation and make sure they receive what's most useful to them. And again, just a final thought on this topic, that even this idea of segmentation of the populations doesn't have to always be genetic either. This is just another recent example from the literature about other ways to think about the use of therapeutics and segmentation of the population. Again, it's been known for a while that the supplementation of folic acid, in pregnant mothers, is beneficial, not just in autism, but in developmental disorders and neurodevelopmental disorders in general. But there have been studies recently showing that in individuals with autism themselves, folic acid supplementation could be beneficial. And in fact, one of these studies recently reported that within the group of individuals that receive the supplementation, a much greater response was seen in individuals who have autoantibodies against folic acid, with the idea that essentially, potentially one of the things that could be impacting the development disorder in those individuals were the fact that you're getting these autoantibodies sort of taking the folic acid out of the system, withdrawing it so it's not there to help, you know, with that early development, but by putting more folic acid into the system, you can overcome it, and those individuals then would be most responsive to that type of therapy. So again, this idea that with a more targeted trial population, you're more likely to see the positive result, more likely to get that positive result, and therefore going forward, more likely to have a population that's going to be maximally benefited by that intervention. So final thought here, I think I've only got one more slide after this. Final thought, just in taking this to the next step and thinking about where else we could take this identification of genetic subtypes of autism. Certainly I talked about, you know, that although we have individual genes, we can round them up to pathways and think about interventions that are a little bit broader, at least pathway-wise, but at the same time, technology allows us to think also about just doing it on a gene-by-gene basis. That recently there has been a very significant resurgence in the idea of using gene therapy, the ability to basically use a virus to reintroduce a normal copy, a functional copy of a gene into a cell where that same gene isn't working. And this is data from some work done with a mouse model of Shank3 autism, Phelan-McDermott syndrome, essentially, where they have an animal that's missing Shank3. And what they've done here is they've actually gone back in the adult animals and put back a functional version of Shank3. And what they reported is that although this animal was born, developed up to adulthood without any Shank3, shows significant behavioral deficits because of this loss of Shank3, that in putting it back in those adults, you actually see a beneficial response. It decreases anxiety, it increases socialization, it decreases repetitive behaviors. That all of these types of inputs all these types of readouts were benefited by simply putting back the missing gene. So it does hold open the idea that what is now being done successfully in humans for things like hemophilia and genetic blindness could potentially be a route for individuals with autism as well, moving forward by understanding what the genetic cause is, having good animal models in place to be able to understand those deficits, be able to test these types of therapeutics, and then potentially take them into humans. So just to wrap up then, final thought is that inclusion of genetic information in clinical practice, I think really could be beneficial, really help us in driving a more precision, personalized view on delivering the care that's needed for individuals with autism, as well as helping them themselves understand better what their own path is. And at the same time, new technologies, new interventions as they come along will both be, I think, driven by some of this understanding, but also some of this understanding will help us actually drive those interventions to a successful registration, to delivery, into the clinics, into the doctor's offices, so that way they can be utilized by those individuals who are most likely to benefit. So with that, I will stop there, and we can take any questions if there are any.
Great, thank you so much. We do have a couple questions, and then folks can always either raise their hands to ask questions or type them into the questions bar if thoughts arise. So the first question is, if you can just talk a little bit about what distinguishes this AS genetic data from other genetic data that's being collected for autism research, and then if there's any collaborations with any of the other individuals who are also collecting genetic data for autism research.
So let me start by saying, one of the ways that we were able to get this missing sort of up and going so quickly is the fact that we've been able to make use of a lot of the investments that have previously been made in actually bringing together well-phenotyped cohorts, and actually that have powered some of the early genetic studies. So for instance, we have several thousand samples here from the Autism Genetic Resource Exchange, or AGREE, that Autism Speaks has sponsored in the past, the Baby SIBS Research Consortium. We've been working with EU AIMS to be able to sequence their samples. So a lot of these different cohorts that have been collected, that have been funded over the years, this is making use of that data. So we didn't go out and have to recollect, you know, and bring in new individuals. That all this money that already been spent, let's utilize all these existing data sets to be able to generate that next generation of data. At the same time, I think the other big genetic study that's out there right now is SPARK, funded by the Simons Foundation. One of the things is that first, that does have a genetic component, but I'd say you know, my understanding is at this point the much larger focus is on it as a registry, and that when they when they are getting into more of that genetic analysis, the focus is also going to be just on the exomes. And again, I discussed early on the difference between whole genome and whole exome, and that our focus from the very beginning, I was doing whole genome because we think that provides a much more robust data set and a much more evergreen data set than just whole exome data.
Great, thank you. And then I think you touched a little bit on this next question, but the next question is, what is the added value of including the good phenotypic data along with the additional genetic data?
Oh, yeah, so I think this is really, so I think there's a couple different advantages to having that really deep phenotypic data to go along with it. One is, I think, just to be able to understand the role of the genetics, to be able to do that genetic to phenotype correlation association. Certainly one of the things we're interested in doing, as I said before, is understanding, you know, specifically the role of each of a particular gene in the phenotypic outcome, but also there's interest in understanding, you know, a broader classification of mutations and outcome. For instance, you know, one of the other analyses that we did within the Nature Science paper was to actually do that kind of analysis, looking at the fact that when an individual has a copy number variant within a autism-associated risk gene, that in fact those individuals tend to have a more significant impact on cognitive ability and development than those individuals within our data set that didn't have an identified variant or copy number variant within those genes. So again, we can make both very specific understandings about comorbidity risk on a gene-wide basis, but also just in this general class, that if you do have a more Mendelian form or syndromic form of autism, what it looks like versus what we still call idiopathic. And I think that's what some of the things we want to continue to do. One of the things we're exploring with Verily right now is to use their computational power to do more of these genotype-phenotype correlations, to be able to get to, you know, to essentially replicate that study that was done with Harvard with the electronic medical records, but now begin to integrate genetic information. And does that result in different subtypings, and what does that look like, and what does a full integration of the whole data set say about the true sort of different kinds of autism, recognizing that really we are talking about autisms and not autism.
Great, thank you. So we don't have any other questions at this time, so we can wrap things up just a few minutes early, or if you had any last thoughts before we sign off.
Well, this is Dan. I'd like to make a couple of comments here. One of the things that many of us are hearing from insurance companies across the country who want to deny doing the chromosomal microarrays and more sophisticated genetic testing, the pushback from payers has been, what are you going to do with this? And a few years ago, we would have been saying, well, we find these copy number variants, these small differences, but at the time we didn't know what they meant. I think Dr. Pletcher presented a couple of really nice cases today of where we are seeing and identifying specific findings that may result in a change in the clinical management of these types of autism. And as he was emphasizing, we are looking at this much more now as autisms. If you will, he presented the CHD8 type of autism, for example. And in the slide that he had regarding circadian rhythm patterns and sleep problems, those are a couple of other ideas of where this increased awareness of a genetic underpinning of this problem might, in fact, impact on our clinical management of these patients. So we are getting there. The question regarding phenotypic associations, if we don't have that phenotype to match to a specific genetic finding, then we're not able to classify them. So we need to continue to do both of these as well as we can so we can characterize these different types of autisms. So wonderful presentation, Matt. Thanks very much.
Thanks, Dave. And actually, you know, I wanted to follow up on your comment about the problem with reimbursement around diagnosis, because you hit the nail on the head, and it's kind of disappointing in some ways. And just an anecdote along that line, you know, with a lot of this work, obviously you would imagine the diagnostic companies are interested in what we're coming out with. And we were sitting down with one company that has, you know, I'd say one of their primary focus areas is on a diagnostic platform for epilepsy and related seizure disorders. And they're interested in expanding into autism. And the feedback they were sharing with us is just what you said, Dan, that for some reason they could always get the approval for reimbursement for the epilepsy panel, but they continually get denied for the autism one. And it came from this, I think, misconception where folks felt that epilepsy was more of a medical issue, whereas autism is a behavioral one. And so as a medical one, there was more you could do with that information in epilepsy than you could with autism. But the reality of the situation is that in fact you're pretty much in the same boat in both cases. And in fact, the funny thing is out of the work that's been going on, both in missing and elsewhere, is in fact there are a number of genes that overlap between seizure disorders and autism. You know, SCN2A comes to mind right away, but they're not that different. But there is this sort of misconception that still, I think, holds people back in thinking about autism in the wrong way. That in fact there is anything to do that it's distinct from, you know, the quote medical conditions like epilepsy, when it's not. And then in fact, if you actually had more of this data, it would, I think, progress even further to be able the ability to use this data in medical terms. And so I think some of it is just perception as much as anything else, when in fact the reality is it's just as useful in many of the cases where reimbursement is happening.
Yeah, very good.
Well,
okay. If there are no other questions, I do want to thank Dr. Pletcher for joining us today and presenting this very informative webinar and updating us on the latest progress with genetic understanding of the causes of autism or autisms. We are finished about a minute or so in advance, so I will let everybody pick up an extra